So in order to upgrade or update your toolbox, you just have to visit this documentation page and copy this quick installation code, and then write it in the MATLAB environment, and make sure you have your previous version in the same folder, so the new version will just overwrite the old one. Then everything should be the same otherwise.
Can you show the website again?
Oh, the website, you can Google SCGEA toolbox and read the docs. For students log in late today, and we are just updating the single cell analysis toolbox. Okay. I don't know on your computer whether you have this merged file last week. So last week we use this accession number download mouse long endothelium cell, epithelium cell at a different development stage. So we merged these two data sets and combine them here. If you don't have this data, you may have to use just regular whatever data set you can find as an example data. But because if we start from downloading the data and merge, that will take a couple minutes on my computer. But I don't know if we should do that. How many of you have these data files in saved? None of you. Okay, I can do that again. All we need is this ID, right? So I will use this ID, download the data and save it, and download another, save it. Oh, I can download them all together. So what I'm going to do is just copy them all. Okay, I copy this to ID. If you don't have this, you can type this in. Then I can see, okay, I will select a session number. So you can give one number or two numbers. In this case, we are going to give two numbers separated by a comma. So if you can do this, we are ready. Just recreate the example data I'm going to use. So this is a merge of two data sets. If you still have trouble, you can use just example. You see here in this dialog, I have provided two accession numbers as a example. You can just copy that. Anyway, with two, you will download and merge datasets. I mean, this function should apply to majority of this database record, as long as they provide the correct format of matrix file, either in the txt files or in the MTX. So that's what we are going to do today first. We merge data. Second, we are going to see how we can compute a score. So this score is given by marker genes. So you can define this set of genes, and then we compute the score and assign a value for each cell. So to tell whether this cell has greater value for this score or smaller value compared to other cells. In that case, you can use a self-defined score system to characterize the cells and also compare between them, right? It's very useful, and I will show you how can you define this marker gene set in your own way. So if you have a discovery, say 5 genes will tell you whether this cell is a cancer cell or not. Okay, so you can just build that in this systems, so you can compute the score for every cell you want to examine. We'll do that. Then we'll do a LDA analysis with teapot. Okay, because LDA is to find the angle to look at your data. So 2 different data sets or more can be separate as much as possible. So we still use teapot examples to see how can we separate multiple teapots. In MATLAB, there is a class vacation linear. That's an app. So we are going to take a look of this classification learner. It's a built-in MATLAB app. And what's the functions in that app? So I have some connection issues. I don't know on your computer whether you have the same problem or not. I hope you are okay. So I'm going to use this example data, maybe smaller. So I'm doing the same thing. So if my connection is not okay today. So I got some issues with it. I don't know what's your problem. Do you have any problem in your side? So I will just use the one I already downloaded last week. Yeah.
I also have some errors.
You also? Well, whether that will be an issue with the geo database?
960.
Mm-hmm. It's okay. I don't know why we have that issue. If you still have this problem, I still believe it's a running error, so it's a connection issues. So I copy the link, which in the program we try to get. So yeah. So you see in my browser, and it takes some time. So that's the error come from. Okay, so if you have trouble, you can just use this example data. Okay, just click. That will should work. Right? So this example should work. Okay, I use the data we downloaded processed last week, but you can use whatever the example data. We will do the same thing. Yeah, so we do have an issue with this server. Okay, let's see. There's a new function here, so you can switch the color map. Let's check whether you have the latest version. Then if you do the random color map, you say yes, it will give you a random. But here you should have this option, non-randomly. Then select batch ID. If you have this function, you have the latest version. Okay. Now, there's two batches of data. And then here you see this icon. This button says calculate sales scores from cell type markers. And here you have the similar function also calculate the cell scores from a list of featured genes. They are same function basically, just use different marker gene database. So let's just click this one first. So for this, then you will say, okay, I will have options to calculate scores. Scores are enriched expression, relatively enriched expression of selected gene sets. You can see I can want to select the predefined. So those are the predefined cell type gene sets. Some of this are from papers. So you can see, okay, I look at the macrophage markers get from this paper published 2020. Or I want to see whether I have a late stage of G1 phase cell scores. For example, this one is to compute for T cell exhausting markers. Exhaustion marker, and that's another cell type called fibrocytes, etc., etc. Okay. If I select one of them, I want to try fibroblast in my case. Then this function will compute a score for every single cell. Well, so this gave you the foundation. If you know what markers or what combinations of markers you want to see, and this score will give you the basic information for separate cells into class, right? Yeah, so this is indicate whether fibroblast cell marker genes defined by Han 2020 paper highly expressed in some of those cells. So fibroblast is a cell type, and the Han 2020 study, I don't know what they use, but they defined a particular set of genes to indicate if a cell have highly expressed of those set of genes, the cell is likely to be a fibroblast, according to Han's definition. So this function, just look at the marker genes and look at your data. Assign this relative expression for each individual cells. And what we can see here is that the higher the score indicate, the greater the value of this score computed. And the cells, this group has the highest, so they probably have some properties of fibroblast. Of course, this is only one of the scoring systems. So if you try another one, predefined, you can say, okay, I want to look at cells in the M phase, G2 and M phase. So this is you get another different scoring systems for the same set of cells. So I just put them side by side. Okay. They are. So this scoring, this feature gene set in order to compute the score to characterize your cells, they give you different pictures of the same cell. So if you look at the same, I should rotate this somehow. Yeah, I should rotate this. So those cells, this picture tell you this group of cells are likely to be fibroblast and not in the G2 M phase transition stage. Well, this signature score tell you there's a group of cells here have a higher value, indicated they likely to be a cell in the periphery or prepared to be goes into the cell cycle, or maybe already in because it's in the G2 and M phase transition period. I mean, they're just relative to compared to others, right? Any other questions? So this is a predefined score system. So you have to have a dictionary. So this can be expanded. You can customize this. I will show you how to do it. Well, you can, for example, the immediately customization will just, you define a new score system by selecting some of the cells. You can select, okay, I see. I will select, for example, I know this 2 genes and that 2 genes, 3 genes, this genes will create a new scoring system. So I'm defining a arbitrary scoring system for myself. And you can compute cross cell whether there are some cells have a high expression of this random selected gene set. It doesn't make any sense because I have not putting in any biology. Those are the random genes I selected. So you see the relative expression, it's almost the same, but it's this group have no expression of those combinations of this. Okay, so this gave you a way that you can customize, define your own scoring system in order to look at or whatever you expect to see and to quantify the states of the cell. And this button is the same idea. Well, the markers, the scoring system for this list, there are marker genes for different cell types. Okay. So you can see, okay, I can try this and obtain a set of the system. But this doesn't mean we have a adipocytes in there. Let's just see the relative value for this group regarding to adipocytes marker genes are higher than others. Okay, so this gave you the way, the power you can do scoring system. So I'm going to show you where I define this customized, predefined scoring gene set. So if you look at the toolbox here and click inside, there is a folder called resource. inside the resource, you can find a file called cell scoring txt. You can open it. And the first column is the scoring name. So I just put it in there. And then next are the marker genes, right? It also has positive markers and negative markers in the different columns. And those genes just separated by the comma. Negative markers means if you have that gene expressed in the cell, then this cell is supposed not to be either the cell type you defined or not in the state you described in your scoring system. And at the end, I have this, the last column is the source of this marker reference, okay? So you see, if I can change, for example, cell type, I change fiber size A, so I change the name of this, then the name appeared in my dialog window will also change. So you can completely customize your own scoring system in order to fit your own research and your study system, because in the public domain, maybe nobody will study your cell type. So you want to define your own score system by customize, change this file. It's called cell scores txt file, txt. Okay. Online students, are you okay? I hope everyone knows how this is working. Okay. Remember in the lecture, at the end of lecture, we talk about manual annotation of cells. You want to assign cells into different program or in the states that can describe this relative expression for the marker genes for that program. So this is doing the same, okay? Of course, for the classification problem we have is we need first, I just demonstrate that again. We have done this many times already. I will cluster those overclassed cells into many small clusters. Then for every cluster, I will compute the not individual cells score, but take them as average and compute a score across all the cell types. Otherwise, it will take too long because I cannot wait to let every cell compute all the score across all the cell types, but I will just compute average for each cluster. So that's, I only have 50 clusters, so I can do this very quick. That's why you can see this automatic annotation will just very quickly assign those most likely cell types into as a name of each cluster. Okay, once you finish this, separated clusters will be merged based on their name. And then that's the annotation. So what happened if you don't select automatic? So I will do that again. So if I, in this case, I'm going to set up 10 major clusters. Let's save time. Then, so in this case, if I don't assign clusters cell type automatically, I can select a manual. In this case, I will be, see, if the mouse, okay, there's only options is fibroblasts, so there's nothing else. Oh, that's I have to assign this manually. I think that's there's a problem with my program. Suppose you should have a secondary options, third best guess. So for some problem, I don't know where the problem is. Only gave you the top, the best solution selection. But I should have a updated version so you can fix that problem. It's supposed to give you top 5 or top 10 candidate, which the second one, in some case, can be better solution for compared to the first one, although it's not supported by the computation. Okay. So we are done with this. Done with this. We can also look at deep learning. Toolbox. We will not go deeper. This is just a product in machine learning toolbox, which is a part of the function provided by MATLAB. If we still have time, we will do this DB scan example density based. Class string example. I'm not so sure we will have enough time to do this. At least we will be able to look at the deep learning. Now I'm going to jump to number 4 first, and then because that will be just okay, we can finish this first hour. So I'm going to jump to number 4. We are doing classification learner. Okay, jump to here, then we will go back to 3 to do LDA with the teapot example. So I close all the window. Now all the windows, clear my space. Now I need to load a data called let me see. Yes, so this is a data set which provided by MATLAB as a demonstration data set, and that's a fishery data. So I just read this table in, and you should be able to do the same here. Okay, our job is to use 4 parameters provided by this training data set and predict the species of fish, 3 different sets of fish. They provide the length, width, and the petal length, petal width. So 4 different parameters. And 150 samples. Okay, how can we look at the data? First, I will see. So our goal is to set up a classifier. So can do the classification if I gave the parameters, and I will tell you whether this is a which kind of fish it is. Okay. So I will first do a unique. So from this data table, I see species dot. Okay, so if you put it, so this data is a table, so the data dot species, that's variable name. So this will return a list of fish name. Okay, so if you use unique, and you will get unique set of this. There are 3 unique species name. Okay, I just want to know how many different type of fish in the data set. So what we can do now is to plot the data out. Group. If I use first 2 PCs, not PCs, first 2 variables. See, I will have data set length, data. Okay, so that's one way I'm going to try to color code those data points. Remember, you don't have to type this. Once you put a data point, then you use tab, and you will be able to give the selection of the available. So I'm doing this tab, and you can select the variable names. Okay. So it looks like it's easy to separate this red fish from green and blue fish using this variable length and width. but not so easy to separate another blue and green. So let's try if I do title wise. Let me get some different pictures. Yeah, I select another variables to do it. I don't know whether we have a 3D, but we don't have a 3D group, so we have to use this. Let me set X as this. So I'm doing the 3D plot right now. Then I need a Z. In this case, I can use 3-dimensional X, Y, Z, and data species. Try this. This is the size of the data point. I convert this to string, maybe it's working. I make the point bigger. This is a function to change your group variables into an index. So if you label 1, 2, 3, something like that. Insert it even bigger. Okay. So with this data set, we are going to use classifier classification linear. Okay. Again, you don't have to type the whole thing. You can use tab and then select from the options. Okay, so we have our data, so we know what are the predictive variables, what's the outcome variables we want to set up. With that information, you should be able to see, okay, I want to start a new session for this and using a variable from the workspace. So we can select this workspace, and now we start to define our classification problem. And it says, okay, where is your data set? We will select our data is X, no, it's data itself. Okay, so the program, the app just recognize this species, maybe you use as a response variable. And you can select, if you decide to select others, then it's possible. Then you use the rest of them as a predictors. Okay. Here you can select the cross validation, how many folds. So normally people say 10. You can increase this 10-fold cross validation. Cross validation means you will remove some of the fish and use the rest of the fish to do the training and then predict whether you can use the trained classifiers to predict correctly your tests. And you repeat this for 10 times. You predefine this right now, everything is set up. You can start the session. Are you okay? Do you need time to load up this? Are you okay? Okay, so this looks like a very busy interface. The reason is you have many different classification algorithms already built in this app. So that's why you will find all of them basically. Remember, we mentioned linear discriminant LDA. You can also select the quadratic. So some nonlinear in there and logistic regression. But in our case, we have 3 response possibilities, so it's not applied here. And you have naive Bayesian. You have SVM, KNN nearest network. So there are many classifiers, algorithms you can select from here. Then you can select all, or you can select all the linear, and then you can run them all and see which one is the best. So what's our goal here? So we provide the learning and the training data like this, and then in here you select all the different algorithms and then train the data. At the end, you should be able to export your classifiers, which include the algorithm and the parameters. And it's already trained, the parameter has been set up to give you a best solution for your training data set. You export this as a function. So then you will be able to use it to train, to test for new data set. So that's the purpose of this. in the middle, you first have this original data set and then look at, it's like what we did using command line there, and this is already predefined here. So you select this 2D scatter plot by either the combinations of these parameters, okay? These variables. And 3 different kinds of fish, color coding. Okay. Now, we haven't selected model yet. If I select this, okay, my active model will be showing in this, show in this panel here. So tells me I select linear discriminant algorithm with 4 features. And features are the variables. I want to use them all. We can pre-select features. So if you think some of those are not very informative, you can leave it out and only use 3 to do it. But in this case, we use them all. So features are the variables. We can also run PCA first. But if you enable PCA, then you may summarize your features. Then maybe you can use PC for the. I don't know exactly what they set up with doing here, but okay, so you can have this, and the scatter plot is to show the data points like this. Okay, we are ready. We can have a PC included in there and the linear discriminant algorithm. Then we start to train the data, train the classification method. So you can click this, and also if you have a larger scale task, you may want to parallel functions, then in that case, you may borrow the full power of multi-strain of your CPUs. But in this case, we don't have to use it. Okay. The first outcome for this is a confusion matrix between 3 classes. Then 50-50 means that all correctly predicted. And this one is 3 was predicted as this fish, but in reality, it's supposed to be this, but predicted as a Virginica. And this one is supposed to be Virginica, but it was predicted as a lesser color. So that's the wrong prediction made. And compute all this true, positive, false, negative, true, false, negative, false discovery rate, whatever, all these parameters here. Okay. For some of this, you may be able to have this ROC curve, but I'm not so sure whether we should be able to do this here. Yeah, so for separate Setosa with this other 2, we can get perfect separation with this model. But if you try to separate versicolor with the other 2, you get some of this imperfect results, and then the area under the curve becomes smaller. So that's another way to look at the prediction results, whether it's good or not, the validation. Okay, I think we can take a 5 minutes break, and you, if you have time, just look at this, play with it, and we will try other models. Maybe we try all the models as an example to show you. After 5 minutes, okay, come back at 11:05. Okay, come back to our prediction results. So you see, it also highlight those data points or fish that hasn't been successfully correctly classified. Okay, so they're supposed to be orange and make yellow, or vice versa. So those mistakes have been marked by incorrect with a cross. Now we can try to select, see, all linear options, then the algorithm that satisfies what you selected, the category you will show up in this panel. So there are two models, two different classification algorithms have been selected. You can run them all and compare what's which algorithm is a better solution. Okay, in terms of accuracy, SVM gave you slightly better accuracy level. So from a 95.3 with a 2 linear discriminant model, and we get slightly improved accuracy to 96.7. This may be just one fish has been assigned correctly by SVM. SVM stands for support vector machine, which is another technique we will discuss next week. So similarly, you can see, okay, I want to use all the algorithm available here and apply to my problem. I don't want to run this because it will take some time. But anyway, so you can apply all available methodologies, algorithms, and let the program to train the model and see which one you the best. So now, for example, we are happy with this algorithm and what we can do next. Now, I'm happy with model 2, and I will select it. I'm going to export my model. Into a function so I can use in the MATLAB. And you can save it, and the deployment means you can save it in some other format, you can get it back. But in this case, I will just export my model and make it ready to give a prediction for the new data. Okay. So this will save a variable name train the model in workspace. That's all. So come back to the workspace here. I have a variable name called trended model. So I can type trend model. I have no idea how to use this, but I can see to do prediction. What we can do with this. Yeah, so it seems to give me this indication, so I just type the information. How can you predict? Okay, I think I know how to do it now. So I will use the trended model, and then I see the predict function. Dot predict function. I just gave in the data. The data is a table. So if I only gave the first, okay, I only gave one data point, which is from the table, the first row, which is supposed to be this. So I will only give it 1 to 4 variable, not give it name of species. The outcome, you see, okay, because this data is only 4 numbers, contains only 4 numbers, okay, right? I gave it to the predictor, then it gave me the species name, which is this Setosa. I can try number 1, number 2, 2 data points. They all predicted as a Setosa. Then number 65, this should give you a different prediction. Okay. So I don't know if it's just take 4 numbers. So I will make up these 4 numbers. If I only give the 4 numbers, I hope it can give me the prediction. No, it needs a table. So I have to make this into a table. No, it's still needed. That's this variable names. So you still have to give it a table. So yeah, so it still has to be in the original format of the table. Okay, so for this table, you can make a new you can make a change. So we have a new data comes in. So here I will make it up 6.7, 3.2, 7.2, and 1.1. So I make a new variable and we'll let it try my number 150. Give me the prediction. So the prediction of this new data is Virginica. Okay.
My model got a different prediction, actually.
Are you putting the same values here?
Yes.
Okay.
Oh, wait.
So
No.
your prediction is not Virginica, you get
I missed the first column where everything mine was shifted.
okay. So I made this up because I have a reference which 150. So I probably, yeah, very close to that. But if I give some other range of the data.
I'm getting Virginica now as well.
Yeah. Okay, so this structure apply to all the different algorithms. So it's a unified interface to give you to train the data and create a model. I think it's pretty good for small problems. Okay, I think we are okay with the classifiers. This application. So I'm going to close this app. Let's see, move on to our next topic. We are going to use LDA. So next one will be this. Okay. The function LDA, I want you to copy it from the PowerPoint slide. So let me clean all this, close all. Now I need a function. So I need a function called my LDA, right? We had my PCA function before. Now I need to create a new function called my LDA. So I'm going to use this to do linear discriminant analysis. Well, the function already in written, which you should be able to find from this drive. I will download the PowerPoint file. For number 7, let me see where it's downloaded. Yeah, classification. Then I will move to slide 32, number 32. Then I just copy all the text here. That's a function definition. I copy this, I select and copy this. I'm going to put in chat. So if you are online student, maybe just copy the function definition from the chat window. Okay, I paste it here. And you will see the name is not right because MATLAB require function name is the same as its file name. So I have to select this and rename it to the file name, which is my LDA. Then I save. So we are ready. In chat, okay. Okay, I have my function. This function will take X input data. Give you the rotated data. Okay, it will compute the matrix and rotate. It also needs another variable. It's label. So this label will tell you in the X matrix if you have 100 data points. A label will tell you the first 50 belongs to classification 1. And then the rest of 50 will be group number 2. So we have to give the label for each input data. Okay, because we are doing classification. Now we will have data from teapot again. So I'm going to load to have my X1, the first teapot data in there. Again, this coordinates has already been provided, so I can just borrow it. Now I'm going to rotate this to make another teapot, to rotate the pot and then also move it to somewhere else. So let's put a label first. So I will label it L1. As 1 times once. So I don't have to times, so this is 1, number 1. And then the size of X1, 1. Okay. So this is a series of indicator as 1. I'm going to create a rotator, a matrix 3 by 3 to rotate my teapot to some angle. You know, I can use to make a rotation 3 by 3 matrix, and also my rotation matrix R1. Okay. So I use my rotation matrix multiplied by transpose of this. So I rotate my teapot and then transpose it back. The reason is the coordinates of this teapot in x1 is given by as a horizontal the row vector, but I need a column vector to show the data points and the variables in the row. So I have to transpose it and then rotate using this rotation matrix and then transpose it back. So this will be my second teapot. And also I'm going to add a value to say add 4.1. So I move. So I rotate my teapot and move to somewhere else, to there, separate apart. Okay. My label for L2 is 2 times this. Okay, now I merge my x1 and x2 together. I merge my label 1, label 2 together. Remember, this is L, it's not A, not 1. So it just cannot use 1 as a variable name. Okay, we are ready, and let's plot them out and then see what we have here. Scatter 3, yeah, we do 3 dimension. First, X, second, X, third, and then my color. Are you okay? This indicate I just ignore this parameter, and that's the size of the bubble, the marker. But L is my label, so I should have 2 teapots here. Okay, I will copy this to chat if you, just in case you don't have that. Let's see, where is my history? I will copy this, all the code so far, so you can reproduce if you don't follow. Okay, so if we don't do anything, we just look at its xy axis. It's just 2 dimension. That will be the one we can get from before doing any LDA separation. Now, so that's the way so far we can separate them. The LDA will help you to find a new angle to give you the top 2 PC to define this new coordinate system to separate them better. Okay. So we are going to run our built-in function. The function we just get is my LDA x here. Our label is here. Then this is the y is our LDA results. Now we have y1, y2, y3. Remember to have a figure in front so we can create a new figure. Okay. On your left is original before rotation, before apply LDA. On your right is after. Still XY. So that's the LDA solution. It found this particular angle. It believes that your point is separated the best. If you don't want to manually rotate this, you can use view. And the 2 that will give you the 2-dimension view of your plot. Okay, so that's this should also work if you have a 3 teapots here. We just add 1 more cluster groups. Have more is still okay. Okay, so this source code is available if you want to try later. Okay, any other questions? Still working. And also maybe you know, or maybe you're not, you can have a color map to see I want to set different color combinations for those indications. So by default, that's the default. If I see there are other options, you can see when you tab it, you can have a flag. Jet. Okay. There are different options for table. Those are the many available combinations of the color sets. If applied to active window, so you can still have the original, but once you active this window, now this will apply to it. So you can see agree. Okay, oh, it's gone. So that's different set. Yeah, for some reason, this the default, so I can change it. Okay, any other questions before we move on to next topic? So the next topic we will have a quick look of this deep learning toolbox. We're not going to do anything, just browse its functionality in there and then see, yeah, maybe I can play a video. Deep learning toolbox. Yeah, I think I just play this video and you get some idea what it is.
Deep learning toolbox provides algorithms and tools for creating, training, and analyzing tools. You can use deep learning for image classification and deep learning with LSTM networks, time series, and slowness data. Deep learning toolbox comes with numerous pre-built examples of methodology, object sensing, and technical facial features for regression. You can also build advanced network architectures like DAGs and scientific networks, using custom trainings, shared weights, and Release 2028 introduces Experiment Manager app to manage multiple deep learning experiments. You can track train parameters, analyze results, and compare code from different experiments. You can evaluate training models using visualization tools such as plots and distribution. If you're building a model, that's correct. These models design slice the layers of dimensions and add vertical. After designing and analyzing the network, you import the data, check the data, set training options like learning rate and methods, and train the network all within the app. Afterwards, export the network to the workspace or generate its corresponding MATLAB code so you can Deep Learning Toolbox supports interoperability with other networks. Also import networks and network architectures from the pairs cafe. And since Deep Learning Toolbox supports a model model import models, leverage MATLAB for tasks like visualizing network, then export the model to the speed of training, workstation, or scale the cluster in class, including NVIDIA Cloud and Amazon EC2. Deep Learning Toolbox can be used in conjunction with code generation tools, enabling you to this auto-generated code provides specific performance. If you want a smaller footprint for your network, such as 28 enables you to perform a combination of for more information about the toolbox, please check out the page and don't hesitate to contact us.
I think for us, we just need to know their deep learning toolbox is there, and for our to use. Until you have clear idea how can you apply deep learning in single cell study, then you should be able to borrow those built-in functions and the interface to leverage it, right? Like, we would like to hear your idea about how can you use deep learning to do single cell research. So far, we used altered encoder to build a gene regulated network, represent data and denoise. But that's autoencoder is not a deep learning; it's a shallow. You shrink your neurons, number of neurons to a small set to encode your information, then expand to decode. I don't have any good examples top of my head from deep learning and applied to single cell. I don't know, but it could be. There's potential there. I think for us, we just have some superficial impression of what they provide here. So you have this all the different types of neural networks. And I believe they do have an interface, the deep neural network designer. Let me type that in, say whether we have this in the system. Looks like we indeed have this installed. So let's just bring it up and look at the interface. Okay, so looks like it provides some template, so you can not start from scratch. You have a predefined, pre-trained network structure. You have these different layers of input, different types of this. Okay, it's a really okay. I'm not going to do anything, but I just show. Okay, you have activation function options, right? We talked about also talked about the convolution layer. All right, to me it's overwhelming. I have never played with that before, but we know it is there, and I get it improved all the time as well. So that's a resource, I believe. I think another problem is a lot of the people compliance is MATLAB is not free, so they don't have access to this. But we are fortunate enough, and in a safe environment here, and in the big university, have all this resource, and the documentation is there. So we should take advantage of this, I believe. And also for single cell G expression 2, I developed this standalone package in the GitHub. Again, this is also to solve the problems that a lot of people don't have access to MATLAB. So I make this toolbox available for those people. What I did is a package into a standalone application, so you don't have to have a MATLAB. Any Windows machine should be able to install this and play with that. So I make this available for people. And if you have any colleagues or friends maybe interested in this, please let them know. So I want to distribute this to people who don't have access to MATLAB. I don't know if Google has already have this, but let me try Google, see if we can find. No toolbox, just this tool. So this is a new website, so I don't think Google has already picked it up. But the address is just scga2, that's single cell gene expression analysis tool.github.io. Okay, that's all I want to share this week. We don't have time to visit the last example in the density-based DB-scan clustering example, but the source code is inside this folder, in the class folder, remember? You can unzip this and try. There are a couple of functions pre-defined already, and play with that. You may be able to play with the examples for your homework. Your homework is to explain that paper. I think their implementation of the source code for that paper method to select the outlier points. Okay, that's all. And any other comments, suggestions?
I did have a question about that homework paper. I still couldn't access it. I don't know if you saw the comment, but I did find I found a PDF that had the same title and the same author. I just wanted to make sure it was the same correct paper with all the same
I
content.
update. So I realized the link wasn't right. So I already updated the link. So if you try again, you should be able to access the paper. It's a science paper, and with the same title, it should be okay.
Okay, it was about, I think, 3 pages or so. Yeah, I still can't access it.
The PDF you posted is correct.
Okay, hey,
Yeah, the PDF, yes. Okay, great. Thank you. See you next week, Tuesday. Have a good weekend, everyone.